Alright, here's the final part of this first lecture on microbial metabolism, focusing on the fundamentals. So as we were talking about oxidation reduction reactions, we talked about the fact that in every oxidation reduction reaction, every redox reaction, you have an electron donating molecule and an electron accepting molecule. The electron donating molecule donates electrons to the acceptor, and in so doing, the donor becomes oxidized. So similarly, in accepting electrons, the acceptor gets reduced. So I want you to think about aerobic respiration. So glucose plus oxygen are converted to CO2 and water. The donor is glucose and the acceptor is oxygen. So look here and here. Okay, so starting at the top, we have CO2 and glucose. They represent a couple, a redox couple. Glucose is the donor, and it is the reduced form. CO2 is what glucose becomes after it gives up electrons. So by convention, every redox couple in the redox tower is written with the oxidized form on the left and the reduced form on the right. Okay. Now at the bottom, we've got the oxygen and water couple. Again, the oxidized form is on the left, the reduced form is on the right. So during aerobic respiration, glucose gives electrons to oxygen, resulting in CO2 and water. So let's consider some of the numbers in this table. Every redox couple in the table is organized according to its redox potential, the potential associated with that particular couple. So the redox potential for the CO2 glucose couple is very negative, right? It's up here. Whereas the redox potential for the O2 water couple is very positive. It's down here. Okay, so it's important to remember this. In nature, something can only act as an electron donor if it is giving up electrons to an acceptor that is part of a couple that is lower in the redox tower. In other words, that has a more positive redox potential. Okay, so glucose could never give electrons up to sulfate. Okay, it can't push electrons uphill, so to speak. Glucose can and does donate electrons to oxygen. Okay, that is thermodynamically possible, and there's also a very big drop associated with that. So the delta E value associated with aerobic respiration is quite large. The delta E value will be large and it will be positive. And because of that, the corresponding delta G value is very large and very negative. And it's a very good way for cells to get energy. So you already know a lot about aerobic respiration, but let's think about just applying the redox tower to some other questions that might arise for you. Let's say you were looking at a microbe and it was found in an environment that contained ferrous iron, Fe2+, down here at the bottom. Okay, and you wanted to know, in an aerobic environment, okay, could that act as an electron donor? And could a microbe potentially gain energy from that process? In an aerobic environment, there would be oxygen. So because the oxygen-water couple is below the ferrous iron couple, that's a potentially possible reaction. There's not much of a drop there, so you would want to do the calculation to determine the delta G, but it is possible. Other possibilities are shown here as 1, 2, and 3 in black, red, and blue respectively. So let's look at this one up here. Here we've got H2, hydrogen gas, that's the reduced form. It is donating electrons to fumarate, an organic molecule, as an electron acceptor. So here the donor is H2 and the acceptor is fumarate. There's a very large, very positive delta E value that's going to be associated with that electron drop, and therefore that's a really good way for a microbe to make a living. And in fact, many microbes do use that process. So the redox tower is a convenient visual that we can use to figure out what combinations of molecules could theoretically serve as electron donors and acceptors that are found in a cell's environment, and that would help us make predictions about microbial growth in different environments. I apologize for the blurriness of this. I took this from an actual paper. It's a study of some microbial mats that are found in a marine environment, and the people doing this study were looking to see which microbes were found at which location in the mat and how the time of day affected their metabolic status. And I'm showing it to you from the point of view that I would like you to connect some of the theory we've been talking about with some actual microbes in the environment. So the point in this whole lecture is to start to give you an understanding for how thermodynamics connect to metabolic diversity. And so here we have listed in this table a bunch of what are called dominant physiologies in this particular marine mat environment. We've got oxygenic and anoxygenic photosynthesis, and we're not going to talk a lot about photosynthesis in this class, but we've also got sulphide oxidation, sulfate reduction, fermentation, and methanogenesis. If we look at sulphide oxidation here, we've got H2S and oxygen. The H2S is the donor, okay, and the oxygen is the acceptor. And these are organisms that are able to use that pattern in order to make a living. If you look for these couples in the redox tower, you will find them, and you'll be able to apply the theory that I just discussed on the previous slide and see that these are thermodynamically possible reactions. Similarly, sulfate reduction. So here we've got an organic molecule, and we've got sulfate. So in red we've got the organic molecule is the donor, okay? The sulfate is the acceptor, and the resulting products are shown here. These are thermodynamically possible reactions happening in this mat, and they're done by a variety of bacteria that fit into a large family called proteobacteria. Down here we've got fermentations, so we've got an alcohol produced in here, a variety of fermentation products, and we've got glucose feeding in here, so fermenters are able to use internal electron acceptors, which I'll discuss a little bit more in another lecture. Here's methanogenesis. Methane is being being produced in this particular reaction. There's hydrogen's the donor, CO2 is the acceptor, methane is the reduced product. So we see all of these physiologies in nature, and we can do the calculations and determine theoretical ATP yields given the concentrations of chemicals that they have in their environment and make predictions about growth, which can be very useful in a number of ways. I am an environmental microbiologist who focuses and specializes in how contaminants in nature are degraded. And people like me use this theory a lot. So if you have a plume of contamination that gets into groundwater, a place where oxygen is limited, then what you will see over time, if the contamination is an organic molecule that can be used as a donor, so the contaminant is the donor, maybe it's something like oil or gas, okay, then what will happen when it first hits the water is that the first thing that will be utilized as an electron acceptor is oxygen, because it's the best electron acceptor in nature. Notice its position at the bottom of the tower on the previous slide. The next electron acceptor that will be utilized is nitrate, and then ferric iron, and then sulfate, and finally CO2 under certain conditions. Okay? But we'll see the microbial communities shift as they burn through electron acceptors in their efforts to consume the donors that have been spilled into their environment. We've talked about redox reactions, and we've talked about electron donors and electron acceptors, and I want to finish this part of the lecture by taking these ideas back to what happens in the intracellular environment. So in the intracellular environment, that's where these redox reactions are happening. So in microbes that are using some of the reactions we've talked about, they bring in donors and they bring in acceptors from the external environment, or they use them right on their surface. But they don't do the redox reactions that we've looked at in one big bang. For example, if you took aerobic respiration and you executed it in one single metabolic step, that's actually a combustion reaction, and it would yield all that energy immediately, and the results would be lethal to the cell. So instead, the redox reactions in cells are broken out into multiple smaller components. And the next set of lectures is going to focus on aerobic respiration and what the different components are there. In order to make that work, we have to have biologically relevant electron-carrying intracellular molecules. We call them electron carriers or coenzymes quite often. And they are important because they transfer electrons from donors to acceptors within the cell in a sequential manner. There are two types of electron carriers that are found inside cells: prosthetic groups, which are attached to enzymes, and coenzymes, which are small diffusible molecules. And the coenzymes that you're most familiar with are likely NAD and NADP, nicotinamide adenine dinucleotide and nicotinamide adenine dinucleotide phosphate. Two of the common and really important coenzymes that you've probably already learned about are shown here in this redox couple: nicotinamide adenine dinucleotide and flavin adenine dinucleotide. And you'll notice that their positions are somewhat intermediary in the redox tower. So when NADH, which is the reduced form, is carrying electrons, it can drop them off to an electron acceptor that is lower in the redox tower. So take a look down here and take a look at these areas: cytochrome B, ubiquinone, cytochrome C, cytochrome A. Okay. These are components of electron transport chains, which I'll be teaching you about in the next set of lectures. NADH is able to take electrons from one part of the cell off to the electron transport chain embedded in the cytoplasmic membrane of prokaryotic cells and drop them off. And the only reason it can do that is because the NADH has a more negative redox potential than the components of an electron transport chain. Similarly, FADH is able to drop off electrons to donate electrons to acceptors that are components of electron transport chains. And that's a fundamental idea that I want you to keep in the back of your head as you learn about electron transport chains at that section of the next lecture series. That concludes this first lecture on the fundamentals of microbial metabolism.